Right, so we're going to get started. Oh, have to turn off the sound. Sorry, some hybrid technical difficulties. So maybe if I could just get an indication from people online, can you hear me?
Yes.
Yes.
I can't. Am I getting any thumbs up? Yes. Okay, great. All right, so I'd like to welcome everybody to our Illuminate session today on expansion microscopy. And it's nice to be able to have a lot of you here in person today. We look forward to socializing over some wine and cheese afterwards as well. We're also happy that some of you can join us online, and we're going to record the session so that we can share it on our ABIF YouTube channel. So Natalie Woo is the first-year master's student who's just started working in my lab this September. She was working over the summer as well. And we're embarking into expansion microscopy, which we thought was a tool that might be of general interest to people using the ABIF resources as well. So I'll pass it over to Natalie. Share. Oh, I see. Right. Is it working? No one? No. Yeah, good.
Okay, so hi everyone. Good evening, I guess. I hope everyone's enjoying their week so far. Almost there, almost at Friday, one more day. So yeah, as Claire said, I'm going to be talking about expansion microscopy and how it can be used for super resolution imaging. So I guess just a show of hands, how many of you have heard about expansion microscopy? Okay, few. Okay, and how many of you have tried it before? Okay, all right. And I guess for the virtual audience, if you want to put it in the chat as well. Pardon?
I can put it in the chat.
Yeah. Okay, so yeah, today, I mean, like Claire said, I'm a first-year master's student, so I'm really just getting started on this, and I am by no means an expert. I will be completely honest, tried it once and it worked successfully. So I was very happy about that. But yeah, so I'll just pretty much be giving you an overview about what this tool is about, how it works, and what, I guess, resources you can reach out to if you want to, I guess, one, get started, or you have some experience, want to share your experience with the other people in this the expansion community, or you just want to kind of learn more about what everyone else is doing. So yeah, like I said, I'm going to be first giving you an overview about what expansion microscopy is, and then we're going to talk a bit about applications, what people commonly use it for, or what different variants there are. And then lastly, like I said, the resources that is currently out there for different people of different levels of expansion. So expansion microscopy is a novel microscopy technique that enables nanoscale resolution imaging of preserved tissues or cells using conventional diffraction limited microscopes. And so this technique was developed by Ed Boyden's group at MIT in 2015. And since then, a lot of different labs have put their own spin on it and done a lot of different things. But the way that I like to explain this to people who may not understand what that means is, let's say I have this sample and this bug is what I want to study. So we can see it, but we want to look more deeply into the proteins, look more deeply into the interactions that happen within this bug. And that's where we employ a microscope. And as you may all know, there are so many different microscopes out there, and there's so many different techniques. And I guess what we're always trying to see or do with microscope is, like, how can we look smaller and smaller? How can we get a really expensive microscope to look at a single DNA or RNA? But why not just make our sample bigger and make everything that we're interested in bigger so that we can look at it with, I guess, more conventional microscopes? So expansion microscopy allows this physical isotropic expansion of the specimen prior to imaging. And so you can link these biomolecules or like fluorescent labels as we normally do to your sample. And you link this to a dense expandable polymer matrix so that once you expand it with water, you can allow for this 3D expansion by a 4.5-fold linearly, which is about like 100 times in volume. Or there are other groups who have even tried a greater expansion, but at that point, your sample becomes very, very much like just water, and it's kind of like using chopsticks to pick up water. So it's really difficult to do. But yeah, the most common one would be the 4.5-fold linearly. And that itself already provides you with a lot of information if you're looking at it with conventional diffraction limited microscopes. So this is pretty much a general workflow for how all expansion microscopy experiments go. Like I said, you first attach anchors to the biomolecule labels. And so here in the pink are the anchoring agents, and then the biomolecules are what you're interested in. And then you do a polymer synthesis. So that's the next step here, where you're synthesizing this polymer network to hold all of the structures of interest together. And then you do a mechanical homogenization, or also known as a digestion step, where you pretty much get rid of all the tissue or cell structures that you're not interested in. And then doing this well will allow you to use the water to expand your specimen isotropically. So a major benefit of expansion is also that it clears your sample so that even when you have a really big volume or a sample of a very large volume, it clears it so that the light doesn't get diffracted within the microscope. And so you may ask, I'm sure many of you guys studied different organisms or just different things, different structures, different tissue types. So what can you use expansion microscopy for? So the main uses, at least from what I've read in the current literature, is mainly for studying diseases and diagnosis. Another one is neuroscience, mainly in neurocircuitry, because within the brains, you want to look at a lot of different molecules and how even synapses, like you're able to use expansion for that. And then developmental biology as well. So some people have even expanded whole embryos. And so personally, for me, I'm interested in studying brain tissue, so in mice models. And for my project, I want to pretty much look at the vasculature and look at how these mice models that have brain metastasis and how these cancer cells spread. Yeah, pretty much that's it. And then here are some examples. These are not what I've done, but what I've read other people have done. So I thought it might be interest of you guys. So the first here on the right is E. coli, immunolabeled for membrane lipopolysaccharides. And this was taken with a wide field microscope. So as you may all know, like wide fields are more accessible to your everyday lab in comparison to confocals. So the top here shows pre-expansion and bottom shows post-expansion. So as you can see, it is much clearer, and even the fluorescent molecules have separated so that you don't just get a large blob of light. And then here on the right, you can see this is known as XPath, images of different human tissue types. And so, yeah, you can pretty much study most things like the prostate, lung, breast, pancreas, colon, kidney, liver, ovaries. So these are very expandable tissue types. Some people have suggested trying bone. That would be very interesting. It would be very difficult because of, it's just not a soft tissue. It doesn't seem very easily expandable. But if you can, maybe someday that'll be possible, because if you can link your biomolecules of interest and then kind of homogenize it and still put in that polymer matrix, it should still be physically possible to expand. But, and then here, for those of you who may not be so interested in studying different types of tissues, you can also, so at the top, there's XFish. So I'm sure you're all familiar with FISH, which is fluorescent in situ hybridization. You can kind of merge that with expansion. And so here you can see the post-expansion image in H, where it's a wide field image of the mice brain slice. And you can see the YFP protein in magenta, the mRNA in cyan, and the GAD1 mRNA in purple. And then here on the right, I pretty much shows a confocal image. So now you've kind of advanced your microscopy equipment here, and you can look further. That shows the single RNA puncta within taking up the image from H. And then for those of you who are maybe interested in study developmental biology or work with zebrafish, you can also just expand the whole larval zebrafish brain, which I found was actually pretty cool. So this image was taken under the same conditions just to show how much bigger your sample becomes. And then these are the different variants. So I've already mentioned XFish and XPath. The most commonly used one would be the protein retention, or PRO-XM, which, like exactly what it sounds, you retain the proteins of interest. But there's a lot of other techniques out there. This is by no means exhaustive. Personally, I found iterative expansion microscopy very interesting in that you expand the sample once and then you expand it again. So you have a polymer within it, it's expanded, and then because the molecules are dragged apart, you can kind of go into those spaces and link another swellable polymer matrix into it and expand it again. And some people have found that, to be very, very useful when they're studying like synapses, let's say. And so now hopefully you're thinking, okay, how can I apply this kind of technique to my research, or how can I get my grad students to work on this? It may be very difficult, I think, when I first started, because I didn't really know many people that were doing this technique. I asked Claire, and she suggested that I start an expansion user group where people of all levels of expertise can join and kind of share their experience or questions. So this is the international user group that we have developed. We're currently at over 70 members from over 20 different countries. And like I said, it's open to all. So we have people who have worked with expansion microscopy since its development in 2015. We also have people who are just starting, like me, who kind of sometimes may ask questions that some people may feel like, what? You should know this, but they're still very helpful, and they'll provide all the expertise that they can. And so we're a very welcoming and helpful research group. We also have invited speakers from across the world because we do these meetings monthly through Zoom. It's really just very accessible to everyone. And we record these meetings so anyone can go back and watch them if they've missed a meeting, or they just, they had a question and then, somebody answered, but they forgot. So we also have an organized Google Doc and forms and spreadsheets for information sharing. So this is so that, everybody within the group can access these documents and they can share different experiences about, like, I guess, fluorescent dyes that they've used and they find that don't work, and or reagents that they bought from this company and they think that, they don't work very well in comparison. Maybe people should stop wasting money and buying it from them and purchase from somebody else. So I think people really appreciate that. And here are just some useful papers that I found to get started. The first 4 kind of teach you a bit more about the different types of expansion microscopy techniques out there. And they do a very thorough job about just kind of describing what is good for each one. And then the last one here is protocols. So I followed this protocol my first time and it worked. So I would say, yeah, it's pretty good. I haven't done the cells. I only did 1 brain tissue, but I was pretty happy with the results. So yeah, thank you. And if anybody has any questions, feel free to ask or email me here. And yeah, thank you, Claire, for this opportunity. And I hope you guys all enjoyed it. Any questions?
Yeah, so I'm still trying to get my head around a little bit, but
yeah,
when I think about expansion, I think about if I'm looking at co-localization of proteins to certain organelles and you're expanding the cell or the membranes, doesn't that cause any rupture? I know you said it was.
Yeah, so the lipid membrane is actually destroyed during the digestion process. So you have to pretty much link everything that you're interested in. before the digestion. So within the
So
jelly.
then how do you know? If you're looking for markers or organelles that are lipid based, are you
Mhm.
screwed?
Like an echo of your sound.
An echo,
It
okay.
goes, you know? You know what I mean?
Yeah.
So you've actually linked all the dyes and.
So you've linked all your markers and everything
To the
together,
leaf
so you're
stalk.
assuming,
So
okay, they should look
the
together.
ER is not there because of the shape of the ER,
Okay.
you know? Yeah.
It's pretty much like a mesh. And
Yeah.
then you kind of have everything that you want there, or like a cast, and then when you expand it.
So all
that's
the
it.
elements.
Yep.
Thank you.
No worries.
Based on what you said, like, I guess, like, what you saw, examples of life survey, or
Yeah, so one of the downsides is you can't use live cells because you have to fix your tissue cultures or fix your tissues. Yeah, it would be very difficult. I think maybe you can try developing a protocol if you're interested, but. Yeah, I think, yeah, I would definitely, I don't think it's possible to do
or
live cell. Not that I know of, maybe somebody in the audience can say something.
Well, I mean, there's logic shop, feel it, right? Okay, yeah. So that makes sense.
Yeah.
I wonder if you had any like insight onto mounting medias. Like, I assume you want to use a non-hardening one, but do you know if the gel is affecting refractive index and things like that? Like,
So I haven't used any mounting media, so I don't, so I'm doing the whole brain tissue and then I put it, I expand like dish that I'm gonna image in.
so it's in EBS.
Yeah.
No.
Yeah, so, yeah, I can't help you, but if you join our user group, I'm sure somebody has done a lot of work mounting. But yeah,
So maybe following up on the mounting medium, is there any way to protect the fluorophores? Because our mounting media usually have something to kind of keep them from bleaching. Do you put anything in there?
personally, or
Or is that something that people do?
I don't, I know some people, so I was actually just reading up before, some people, they actually take it out of the water. If you're immersing it in water, it could remove the fluorophores and that stuff. And so some people actually end up drying their samples after expansion for storage. So they just add PBS, which has salt. And then that salt is what ends up drying it, and then they use it for storage. And this is what the Ed Boyden's group has mentioned in one of their Q&A sessions. And so I haven't tried it, but I thought it's interesting because it was like, yeah, if you can just add some salt and it'll dry up, maybe you can store it for later imaging. But yeah.
I mean, an old lab, we used to make our own mounting medias with in propyl gallate that produced for bleaching.
Yeah.
So
That's what I was saying. Definitely. Yeah, so you could probably add that if you need it.
I'll just ask a question, like, what kind of difficulties do you face? I mean, do you face when you're doing microscopy? Is it easier or is it difficult or?
Just thinking you should repeat the question.
Sorry, could you repeat? And then I think, yeah, you might have to speak a little louder.
You could repeat it.
Yeah, so what kind of difficulties do you face when you're, providing samples or using microscope? Is it difficult or is it easy to use a microscope or?
Oh, like,
What
just,
happened last week?
oh, okay, yeah, so I guess. Okay, yeah, so somebody in our audience asked like what kind of difficulties are faced after expansion and then, handling the sample and then, imaging that. So personally, what happened for mine was I expanded it and then I didn't move it into, or I didn't expand it in the dish that I wanted to, image in. And so I had to move it. And because the sample essentially becomes like 99% water, like I was trying to use a paintbrush, a spatula, I like pretty much pulled out everything that was like had a flat surface in the lab and tried to like get it out. And then I'm gonna show you an image later, but like I tried to take like time pictures of like the time points of like the expansion. And I was like, oh, it looks so great. And then I like kind of tried to move it into the dish and it just turned into like some parts of it flipped, some parts of it like clumped together. And I was like, well, this doesn't look like a brain anymore, but I'll try to image. So essentially the most, I guess, issues people tend to have with expansion is like handling it after, because it literally feels like unsettled jello. So, but imaging itself. So the issue that I also had was, so I had a tail vein injection of lectin 649 in there. And I've read other papers they said like a lot of the infrared ones don't work very well, but I haven't seen anyone use the lectin, and so I wanted to give it a try. And yeah, after I imaged, I couldn't find the blood vessels anymore. So I'm probably going to do some troubleshooting and make sure that maybe it was just the homogenization or digestion step. Maybe I did it for too long, and so I got rid of the fluorophores, or generally maybe that like the 649 just doesn't work with expansion. So there are a lot of published protocols on which I guess dyes and like fluorophores that you can use that are good for expansion, but it really, I guess, depends on what you're studying too, right? Like you need to adapt to that. Yep.
Is it possible that the problem was not the dye, but the fact that it was tied to a lectin? Maybe the lectin did not stick?
Yeah, it definitely, yeah, I was looking into that too. Like it could be, and I know people have said like cyanide dyes they get completely destroyed. And so yeah, I think I'll have to look more into what type of, I guess, what epitopes are retained. So yeah.
And that's Natalie, so you highlighted the problem. What was the solution? Do everything in one? Do you have a special mounting platform that you use where you don't have to transfer, or what?
So yeah, I think for just the handling part for me, I think I'm gonna start doing the expansion in the dish that I'll be imaging in.
Okay, so you have a special.
So I pretty much just eliminate that step of having to move it. But yeah, aside from that, I see a lot of nodding, so I'm sure.
We have very much the same experience with our, it was kind of a mess. They expanded it, but it was incredible.
In the dish, and you're like, okay, great. And then you try to handle it and you're like, yeah, it doesn't turn out so well.
So the downside is you can only like a half the grain.
If you,
It will be the size of the dish.
oh, I
Yeah.
see, so
Also
that's
very
the
low.
technical issue, that's why you did it in the dish in the first place.
Yeah.
Okay.
Yeah, because it grows, I didn't really, it grew so large. I expected it to grow very large. So I like used a big dish. But then that kind of the after problem of, okay, how am I going to image this really big sample now? Also became an issue. Yeah.
And just that size thing is generally going to be an issue, right? In the sense that if you
Yeah.
invent something at some point, you might be outside of the range that your objective.
Yeah.
So if you're using a higher resolution objective, that you'll only get a small part of it. So it's a few different dimensions it seems should go, right? That we're not always thinking about.
Yeah. And I guess one more positive thing is, like I said, it clears the sample. So it also helps with like the scattering.
Because it's clear, you can only, well, you can go SD because it's larger.
Yeah. So yeah. Yeah. Is that everything? Great. Let me check if anyone has a question in the chat. So somebody asked if it's possible to apply the protocol to suspended cells, or do they need to be adherent? So I personally have not worked with any cell culturing, so I don't know if anybody here has. No. So I'm not sure, but I could definitely get back to you.
I think if you could embed them in the gel, I don't see why it might not work.
Yeah. Yeah, I don't know if it'd be an isotropic expansion though, if it's adhered. But yeah, that would be definitely, that's a very good question. I will look into that.
I just put it on.
But yeah, and if you're interested and if you want the link, you can to our user group, you can definitely come by and I could share it with you. And yeah, everyone's welcome to join, even if you're not even doing expansion, like, and you just want access to these kind of materials in case you want for future reference. Yep.
Just a quick question, are the reagents kind of run-of-the-mill stuff off the shelf, or is there anything specific that's really hard to get or expensive?
Yeah, so the protocol itself, they're like, or the papers advertise it as very, they're just your average lab bench materials. But I found personally that one specific reagent, the sodium acrylate, I don't know how many of you here may have received my email, but I was looking for that reagent because it, is out of stock in a lot of places. Yeah, it doesn't seem to be very readily available. But you can probably order. I think the problem that I had was like they couldn't find a supplier, so that it was back ordered for over a year, and I was like, that's going to be half of my master's. So, but yeah, all the other stuff, and I think just they require quite a minute amount each time. So for the most part, you're really using like PBS and like water. The other reagents, thought that I had to use a lot of, but really I ended up aliquoting a lot of it. So it can be used for, once you've prepared your solutions, it can be frozen and kept at -20 degrees for quite a while. Yep. Okay. All right, thank you, everybody. We're gonna move on to wine and cheese.
Yeah, so thank you to Natalie, and I guess maybe in 6 months we can do this again and tell you about the progress, but I just would echo the international user group. I'm always really impressed that these people who got it to work are willing to come and be with the group and tell people about their successes. And the database we're building up with the forums is really interesting because we really want people to share things that didn't work. And I think that, you know, this sort of doing things in a community this way can really help advance things more quickly, since we don't usually publish the things that didn't work. Next month, we're going to have the session. I think it's just the second Thursday in December. I think it's December 8th. And I'm going to talk about the different bioimaging networks in Canada, the US, and globally for information. And thank you to those of you who've joined us online. And we'll put the recording up on the ABIF YouTube channel if anybody wants to pass it along to anyone who couldn't make it today. And we're meeting in room 132 downstairs for wine and cheese. So hope you can come. Thank you.